Hello everyone, welcome back to my channel. So this is part 3 of plant physiology chapter photosynthesis in higher plants. I have already uploaded part 1 and part 2 videos of the quick and concise notes. I'll provide the link in description box. So let's begin with this video. The first topic for today's is photophosphorylation. Now, photophosphorylation is a process of ATP synthesis. It takes place in mitochondria and chloroplast. The ADP, along with the inorganic phosphate, leads to the formation of ATP in the presence of light. There are two photosystems, PS1 and PS2. When these two photosystems work together, first the PS2 and then PS1, it leads to non-cyclic photophosphorylation. And when only PS1 is functional, that is your cyclic photophosphorylation. So let's discuss both of these in detail. First, non-cyclic photophosphorylation. As we already discussed that there are two photosystems. First is your PS2 and second one is your PS1. These two photosystem works in series. These two photosystems are connected by the Z scheme and this is known as electron transport chain. The ATP and NADPH plus H plus are synthesized in non-cyclic photophosphorylation. Both PS1 and PS2 are found in the lamella of the grana. See how this process takes place. First, there is a light harvesting complex from which the electron is excited to an electron acceptor. These electrons lead to the electron transport system downhill the movement and reaches to another light harvesting complex which again excites the electron to an electron acceptor. Here the ADP is converted to ATP and in second electron acceptor the NADP plus gets converted to NADPH plus H plus. So the first part that you are seeing, this one is PS2 and the second one is PS1. Both of these are functional in your non-cyclic photophosphorylation. Next we have the cyclic photophosphorylation where only PS1 is functional. The electrons within the PS2 here are inactive. The location of cyclic photophosphorylation is in the stromal lamella. The PS2 and NADP reductase are absent in cyclic phosphorylation and only ATP molecules are synthesized. Now let's see how this process takes place. 700 as the light harvesting complex, the electron gets excited to electron acceptor followed by the electron transport system. It reaches back to this P700 and ADP in Pi is converted to ATP. This is your PS1, your cyclic photophosphorylation. Next very important topic is your chemiosmotic hypothesis. It is a mechanism of ATP synthesis in which the photosynthesis is used for the production of ATP synthesis and this is linked to a proton gradient development across the thylakoid membranes. How this happens is that first the protons get accumulated in the thylakoid membrane in the lumen and then the ATPase has a channel which is responsible for the diffusion of the protons. This diffusion across the thylakoid membrane releases some amount of energy which activates this ATPase which leads to the formation of your ATP. So here is your chemiosmotic hypothesis. Now where is this ATP and NADPH that we have got is used? It is used in the dark reaction which is your biosynthetic phase. Both ATP and NADPH are used to drive the process of your photosynthesis, leading to the formation of your food in the form of sugar. It is also known as dark phase because it is independent of light and it depends on the products of light reaction which is ATP and NADPH2. The location of the dark reaction is in the stroma of the chloroplast. Now the first stable products of carbon dioxide fixation. So the first product in 3 is a 3 carbon compound, that is 3 phosphoglyceric acid in your C3 pathway. And in C4 pathway, the first product is a 4 carbon compound, which is your oxaloacetic acid. I hope till now all the concepts are clear. We will discuss both of these pathways in the upcoming video. Thanks for watching.